Good morning. A pleasure to be here with you. I'm happy to introduce our first speaker. We'll have two short presentations and then a panel discussion. Raju Krishnalapati is the Paul C. Cabot Professor of Genetics and Professor of Medicine at Harvard Medical School and has appointments at Boston Women's and Mass General. A part of his research effort is focused on human cancer genetics and genomics, and Dr. Krishnalapati is a promoter of incorporating the principles of personalized medicine into medical practice. He's a fellow of the American Association for the Advancement of Science, a member of the Institutes of Medicine of the National Academy of Sciences, and currently a member of the Presidential Commission for the Study of Bioethics. Dr. Krishnalapati.
Thank you very much. Great pleasure for me to be here and to share with you some of the thoughts about the current status of the knowledge about cancer and how that informs us about diagnostics, and my views about how this knowledge is going to change the way that we think about diagnostics. So first of all, I have a conflicts of interest. I serve on the boards of these four companies. As all of you know, the current status of cancer genetic testing, primarily for a long period of time, have been testing for mutations that predispose to cancer, and well-known genes such as BRCA1 and 2, and for colon cancer, genes such as APC and DNA mismatch repair genes, and many other genes that are known to be involved in cancer predisposition. And there are a few somatic mutations in a few genes that have been approved as companion diagnostics, and there are some companies that currently offer RNA-based testing for prognosis of particular types of cancers and the efficacy of treatments. However, things are changing, and there is already efforts underway. There are companies that are currently offering large cancer gene panels. They're doing the germline testing or testing for somatic mutations, and Harold already talked about the importance of expression profiling in some cases, and I'll show you some evidence that suggests that testing for structural aberrations is also going to become very important. And one of the wonderful things about diagnostics is that unlike therapeutics, which are increasing in cost, fortunately the cost of diagnostics is going down because the technologies that enable us to be able to do these diagnostics are changing, and primarily driven by reduction in DNA sequencing costs. So let me give you some examples about how things are going. Here's an example that Harold talked about, and this is a poster child for how things have changed over the last 10 years. In 2003, the only type of treatment that was available for this non-small cell lung cancer was chemotherapy, and chemotherapy was effective maybe in about 15% of these patients. And the first targeted therapy that really became available was 2003, when a tyrosine kinase inhibitor was approved, and the drug was approved for all of non-small cell lung cancer patients. And soon thereafter, it was discovered that it is possible to identify patients who have a high likelihood of responding to the tyrosine kinase inhibitors by looking for mutations in this gene, EGFR. And there are only about 10 to 15% of the patients who have EGFR mutations that can be identified. About 25% of the patients have KRAS mutations, and although there is no currently approved therapy for those patients with KRAS mutations, there is clinical data now that show that a combination of MEK inhibitors, a pathway inhibitor in which RAS works, and chemotherapy is very effective in this subgroup of patients. EML-ALK4 a structural aberration that involves in the activation of a tyrosine kinase. There is a drug that was approved in 2011 for patients with this particular structural aberration, and there are about 5 to 6% of the patients that have such a change. There are other types of changes, such as amplification of B2, and there's drugs that are approved for treatment of breast cancer patients with similar types of changes, and there are currently clinical trials that are currently underway to test for the efficacy of those mutations or amplifications in this cancer. And other genes, such as BRAF mutations, even though a small proportion of lung cancer patients have these mutations, there are already a drug that is approved for melanoma patients, and the nature of the genetic change that is present in these lung cancer patients is exactly the same as the one that is present in these melanoma patients and other tumor types. So at the time that this slide was prepared, about 60% of the non-small cell lung cancer patients have either mutations for which there are targeted therapies that are available, or there are other types of changes for almost all of which there are currently clinical trials that are available. So the best type of treatment that one could provide for patients in this category is to be able to test not for one or two genes, but to test for all of these different sets of genes, and to try to determine whether you'd be able to provide them with a targeted therapy or try to introduce them to clinical trials where these drugs are currently in development. So the question is this unique to non-small cell lung cancer? And the answer turns out to be no. This is a figure from one of my colleagues, Levi Garraway's paper in the Journal of Clinical Oncology earlier this year. And it shows that this type of classification is possible not only in the case of non-small cell lung cancer, but in squamous cell lung cancer, in breast cancer, in colon cancer, melanoma, head and neck cancers, ovarian cancer, and glioblastoma. And the current genomic efforts that are supported by the NCI and NHGRI and many other institutions around the world have been doing these large-scale studies, the kind that Harold described, and almost all of those studies very clearly show that all of these different types of cancers can be classified into these different categories. And one of the really interesting aspects about that is that some of the changes that are present are common to many different types of cancers, and therefore it's possible that the drugs that one approved for one type of cancer could be tested for their efficacy in these other types of cancers. So one way that we think about that is that perhaps it'll be easy to consider testing for all of these different markers in one single assay. And from a clinical point of view, one of the nice things about these observations is that many of these genes in which these mutations or other types of changes have been observed or also have drugs that are currently in development. So many of the academic medical centers, for sure, would be capable of selecting these patients for enrollment to these clinical trials based upon this type of information. And I also want to argue there are other types of testing that need to be done. And here is an example of that. Here is a figure from the study of mutation rates in colorectal cancer. And it has been known for a number of years that there are certain subset of these colon tumors that have high mutation rates. And it was thought that many of those mutations are the results of changes in DNA mismatch repair genes. However, it turns out that the examination of these tumors very carefully that is shown in the inset shows that these are the results of not only changes or mutations in genes that are involved in DNA mismatch repair, but other genes such as polymerase E, which is actually involved in DNA replication, is also very important. And it also suggests that the current methodology that we use to detect this group of tumors is inadequate, and we need to be thinking about it in a completely different way. The reason why this is significant is that it turns out that these types of hypermutation frequencies is not only present in colorectal cancer, but also present in endometrial cancer. And one of the remarkable observations that have been made is that those patients that have mutations in polymerase E and that have the highest levels of mutation rates, as many as 1,000 mutations per megabase, their overall survival is extremely good. And those patients that don't have these hypermutation rates, those survivals are not as good. So clearly, the ability to be able to find out this kind of information would help tremendously in thinking about the kind of treatment that these patients can be offered. And obviously, looking at any one gene or looking at the mismatch repair the way that it is currently done is completely inadequate. The other types of things that I want to illustrate to you is that although it's been recognized for many years that structural aberrations are very important in hematologic malignancies, their importance in solid tumors have been underappreciated. And now there is a lot of information that is emerging that shows the structural aberrations also play a very, very important role. Here is just one example of a kind of structural aberrations that are found in colorectal tumors, where it is known that Wnt signaling pathway plays a very important role. But here is another example of a structural aberration that also results in Wnt activation. One of the really remarkable features about this aspect is that it turns out that even though it's been known that Wnt signaling pathway is very important in colon cancer, many people thought that the primary lesion would be in one gene, APC, but it turns out now that mutations or alterations in as many as 18 different genes in the Wnt signaling pathway play a very important role. So if you're thinking about a drug that actually is capable of inhibiting the Wnt signaling pathway, looking at one gene is not going to be sufficient. One needs to look at all of these different genes at the same time. And another example of this, and when you saw the picture about the categories of different mutations that are present in lung cancer, there were certain sets of tumors whose lesion was not understood. And now when we examine them very carefully, it turns out that a whole bunch of those have driver mutations that are really resulting from the structural aberrations. And some of the ones that we've already talked about, like the EML-ALK, but there are other types of genes like RET, ROS, and other sets of genes. And it turns out that some of the drugs that are approved currently approved are also capable of affecting some of these other types of changes. So just looking at mutations as we currently do for many of these things is not going to be adequate, and we need to be able to look at that. So I'll close by saying that here is a set of tumors for melanoma that have been looked at, and you could see all of the well-known genes like BRAF, NRAS, and NF1, and so on and so forth, and all of these sets of genes that are altered. And as you recognize that many of these genes, we also have therapeutic approaches for them. But there are some subset of tumors that are shown on the extreme right-hand side that don't seem to have any of these types of drivers. However, it turns out that when we actually look at them by DNA sequencing, it turns out that many of those have structural aberrations, and some of those structural aberrations are shown underneath, and that involve genes such as AKT, for example, and for which there are currently drugs that are currently in clinical trials. So the point that I want to make from all of these things is that currently the way that we think about diagnosis of looking at one gene at a time and trying to make a clinical decision may not be the right approach to go, and that we need to be thinking about examining large numbers of genes, perhaps all of the known cancer genes, and to be able to ascertain what is the most appropriate type of therapeutic opportunities that they have. Thank you very much.
Thank you, Dr. Kuchilapati. Next, I'm going to introduce Dr. David Solit. David is a practicing medical oncologist and laboratory investigator with the joint appointment of the Department of Medicine and the Human Oncology and Pathogenesis Program at Memorial Sloan Kettering. And his recent laboratory work is focused on the identification of mutational events that co-occur with and cooperate with mutant BRAF and melano. Excuse me, a little allergic in the fall. And abrogate BRAF addiction and thus response to selective RAF inhibitors. His research group has also been active in the development of novel methods to genetically profile formalin-fixed paraffin-embedded tissues for somatic mutations and copy number alterations. Again, we'll have another short presentation and then all of your eager questions.
Okay, great. Thank you. So thanks for the invitation to talk. I'm going to focus not on how we're going to do the testing, but why. And I guess the pointer is no pointer. Okay, just a mouse. Okay. Okay. So really, the focus of my laboratory work is to try to accelerate drug discovery in oncology, and it's a pretty straightforward approach. We looked at defined targets. These are typically mutations or alterations found somatically in the tumor. We look to identify drugs that inhibit those targets, and then the real challenge, and what we're here really about today, is the challenge of identifying which patients have alterations in particular targets so that we can give them a particular drug. And there's two approaches that we've gone about to identify these targets. You're probably going to hear a lot today about this genotype to phenotype approach, where we retrospectively look at large sets of tumors, looking for recurrent alterations, then develop a drug, and then test prospectively whether that drug is going to work. But I'm going to focus in this 10 or 15 minutes on really the opposite of that, which is the phenotype to genotype approach, where we take patients who have responses to drugs in an undefined population, where we didn't have a biomarker going into the study, analyze those patients retrospectively, figure out why they were unique, and then hope to do an iterative study going forward where we do a biomarker selection prior to study entry. And so the inspiration for this was this case. This is a patient who had one of these extraordinary responses on a trial of patients with bladder cancer. And so this was a study we did a couple years back, trial 08123. This was a very simple study design. It was a single-agent study of everolimus. This is an inhibitor of mTOR in patients with bladder cancer. The bar or endpoint for this study was incredibly low. All we were looking to do on this study was to have 2/3 of the patients progression-free at 2 months when you do the first staging study. But despite the fact that we set the bar that low, this was a negative study like every other study we've done in bladder cancer over the past 20 years. But despite the fact that it was a negative study, there were several responders on the trial. There were 2 patients who reached RECIST criteria responses. And of those 2, there was one who's now actually a complete response going on 4 years on this single-agent therapy. And so we asked the question, why was this patient unique? I don't have a lot of time today. We went and we did some targeted sequencing. We did a sequenome assay. We did an array CGH. All of those were unremarkable, but fortunately, next-gen sequencing technology had evolved to the point that we could actually do a whole genome sequence analysis in this patient, and we're able to identify 2 mutations that were the basis for this response in a cooperative fashion, one being a 2 base pair deletion in TSC1, which caused a frame shift and early truncation, the second being a nonsense mutation in NF2. So why are these mutations interesting? Well, both of them, when you lose function of these genes, you get activation of mTORC1, which is the direct target of this drug. And in all likelihood, this patient lacked other mutations that diminished addiction to this pathway. So that was really the first example. I would point out that there was a lot of published data as to why this would be a relevant example. Everolimus actually is approved for patients with tuberous sclerosis. So this is a germline disorder where patients get multiple cancers at a young age. In particular, they get brain tumors called astrocytomas. And notably, all patients, 100% of patients who get astrocytomas in the setting of tuberous sclerosis respond to everolimus. And these patients have germline alterations in either TSC1 or TSC2, hence the name, and presumably lose the other copy within the tumor. Not only do 100% of these patients respond, but they respond durably. And actually on this study, which was published in the New England Journal, only a single patient had progressed within the first 2 years on therapy who had one of these brain tumors in the setting of everolimus treatment with tuberous sclerosis. So it's not surprising potentially that our patient who had a somatic mutation in TSC1, who had the same mutation that these patients have in the germline, responded potentially to the same compound. So we've been able to actually use this paradigm to look at a number of other cases. I don't have time to talk about most of them today, but I'll just mention one other. This is a patient who by September of 2009 had ovarian cancer that had progressed to metastatic disease that was out of treatment options. She then enrolled on a trial through the GOG 239, which was a single-agent study of the MEK inhibitor AZD6244. This was a disappointing study. The response rate was only about 10%, which was lower than they had hoped. But this patient had a complete response by November of 2010, and actually through July 2013, the patient's still on drug, NED, without evidence of disease, and thus really qualifies for one of these incredible, extraordinary responders. Initially, they looked for hotspot mutations in this patient in BRAF and KRAS and did not find any. So we took this tumor and asked, what was the underlying genetic alteration that led to this response, and it turns out that the patient has a mutation in the MAP2K1 gene. This is actually the gene that encodes MEK1, but has a very difficult to detect mutation. She has a 15 base pair deletion, which results in an in-frame protein, which when you generate the construct of this deletion, this is an activated MEK1 construct constitutively, which is still sensitive to the MEK inhibitor. So again, this is not rocket science. The patient got a MEK inhibitor. She has a MEK1 mutation. The question is, can we find these alterations prospectively and do trials of these drugs in patients who have these particular alterations? So this is really the paradigm. We do an unselected trial, a phase 2 trial of everolimus as an example. We see patients who have some of these dramatic responses. We do some genetic assays to figure out why. We do biology in the lab, and this is key to confirm our findings are actually the key alterations. We then need assays that will allow us to find these alterations prospectively so we can do an iterative study where we select patients who, for example, have TSC1 or TSC2 alterations or NF2 alterations and define what is the response rate in that population. So how are we going to do these assays, and what trial designs do we need to do? And I'll focus on that just for the next few minutes. So there's really two main trial designs that would allow us to do this. Dr. Varmus mentioned one of them, which was the MATCH study, and that really falls under this category of genotyping studies. And in that case, you typically start with a disease like lung cancer, where I'm showing an example here, and you genotype those patients or profile them in some way, and then you allocate those patients based upon that profiling result to get different drugs. And so in the case of lung cancer here, if the patient has an EGFR mutation, you give them the EGFR inhibitor. If they have the BRAF mutation, you give them the BRAF inhibitor, et cetera, all the way down the line. Or if they've got no particular alteration, maybe you give that patient chemotherapy. So what are the problems with this approach? And I would just point out three here quickly. One of them is that the drugs are oftentimes not best in class within this study. And so the real best example of this study is Roy Herbst's battle study. And unfortunately, Roy probably had to negotiate over many, many months with multiple companies to put together all the drugs within this study. And by the time he got the study open and was enrolling patients, many of the drugs in the study had already failed in that biomarker-driven cohort that he wanted to test. And a good example was sorafenib, was his potential drug for patients who had BRAF mutations. Another problem is that you often do an adaptive randomization with these studies. That's what was done with Battle. So you start off initially allocating patients fairly randomly, but then when you see responses in one cohort, you might enrich for that particular cohort with that association. The problem with this approach is that it actually can become unethical, or at least very uncomfortable, to allocate the particular arms at some point on the study. So data was coming out that EGFR mutations had a 70% predictive value for response to erlotinib, but they were actually still randomizing patients on the Battle study who had EGFR mutations to sorafenib. So what patient when that data was already public, would want to be part of that adaptive randomization. And then the final point I'll make, and I think this is key, is that this is just not a great trial design for rare mutations. If the mutation's only found in 1 or 2 or 3% of the study, you need very large studies to really be able to answer the questions which you're proposing. And I think BRAF is the best example. BRAF is found in lung cancer in 1%. In the battle study, they only had about 100 patients on the study, so maybe they had 1 or 2 at most patients with a BRAF mutation on the study, and so they really couldn't even ask the question, does BRAF mutation predict for response to sorafenib, because they had insufficient power. So what's the alternative trial design? This is something we're calling the basket. I think you come up with many alternative trial designs, but this is one of them. And this is really the schema for the BRAF vemurafenib basket trial design. So instead of starting with the disease, in the case I showed you before, lung cancer, here you start with the mutation or the gene. And so in this case, as long as you have a codon 600 BRAF mutation and you don't have melanoma because the drug's already approved in that disease, you're eligible for this study. And so we're looking for patients with colorectal cancer and lung cancer and breast cancer and ovarian cancer, rare diseases like Erdheim-Chester disease, which is non-Langerhans histiocytosis. And we always throw in an other category because you never know who's going to show up. And we've seen patients with cholangiocarcinoma and pancreas cancer end up on this study as well. And so you take all these patients, they have the BRAF mutations, you put them in a basket, and then statistically, you're really just doing a set of parallel phase 2 studies. So you're looking for a response rate. And what we've seen already on the study is that not every tumor responds the same way to the RAF inhibitor, even though they might have the same BRAF V600E or K mutation. We're seeing very little activity in colorectal cancer. We've seen other rare diseases where the response rate is 100%. Lung cancer, it's about 40%. Melanoma, it's about 60%. And you can see examples of this which are pretty rewarding. This patient's not actually on the basket study, but this was the first patient we had with a BRAF V600E mutation treated with dabrafenib, in this case, one of the selective RAF inhibitors at Sloan Kettering. You can see by 6 weeks, this patient with fairly advanced disease had a complete response. This patient remains on the RAF inhibitor at 18 months in a complete response. So again, this seems like a very obvious example of what we want to do. We want to give patients with a BRAF mutation the RAF inhibitor and see if we can respond. We just need novel ways to try to deliver and bring that together in reality. So what are the advantages of the basket approach? I see patients, but I also run a research lab, and it allows for testing a defined biologic hypothesis. Do patients with BRAF lung respond, and what is the response rate? Since it's a collection of multiple diseases, you can actually ask that question, are there lineage-specific differences? And if you find them, maybe those lineage-specific differences are due to differences in the pattern of co-mutations. Maybe it's differences in stromal factors, such as secretion of HGF, which can cause resistance to the RAF inhibitor. And you can actually require tissue collection and central review of those tissues to answer these questions over time. What are the primary criticisms of the approach down at the bottom from clinicians or companies or regulators? One is that you may fail to identify patients who don't have the biomarker but could still respond. I agree it's a criticism, but it's not the point of this particular hypothesis-driven study. I would actually say the primary criticism has actually been academic, and I hate to say it. It's been very difficult to get different disease teams to work together. So the breast people want their own study, the lung people want their own study, the colon people want their own study, and that kind of works well for breast and lung cancer, which are common cancers, and usually the drug companies are responsive to opening up studies in those diseases. And really where the basket study, I think, has made the biggest impact is in rare cancers, where it's hard to get studies open in bladder cancer, let alone something like Erdheim-Chester disease, where there may be 15 new patients a year in the United States. So I think this design, especially with that other category, allows patients with rare diseases to get these compounds. So we've spent a lot of time, I don't have time to talk about it today, maybe we can talk about it in the discussion, of trying to rate different genes or alterations as to the level of evidence that those phenotype or genotype to phenotype correlations are going to hold up. And I'll just mention one or two of the levels. Level 1 is where you've got a genotype-phenotype correlation that's 100% established. This is, say, FDA-approved standard of care, and that would be BRAF V600E in melanoma or L858R EGFR in lung cancer. But if you take the same genes and you look at another cancer, maybe that's a level 2 mutation. So what is the likelihood of activity of BRAF V600E in colon? We've actually found out it's very low. So you can't just presume that, and the evidence is obviously less. And then there's other levels of evidence where we have even less convincing data, maybe simply based upon preclinical studies. So just one example of where we've taken this and opened up a new study. This is just this is the results of a retrospective analysis of over 7,000 cancers for either HER2 amplification or mutation. And what you find is that there's a lot of cancers where amplification or mutation of HER2 is found, but at low frequency, in the 2 to 5% frequency. So it would be very hard to do all the studies that would really, answer the hypothesis, where do HER2 mutations, effectively respond to a HER2 inhibitor? Why open up 30 different studies in lung cancer, endometrial, et cetera? Why not just open up one study that would cover all that? And that's what we've done. So here's just the hotspots for HER2. And I would just say that these patients exist. This is a patient with bladder cancer. This is a Foundation Medicine report. You can see this patient has an S310F HER2 mutation. Other co-mutations that may or may not make them resistant to a HER2 inhibitor. What I would comment on is that this patient actually never saw a HER2 inhibitor, actually died of their disease before we could get them a HER2 inhibitor because we couldn't get the insurance company to pay for it, and there really is no proven data that it would work in this setting. But this is not an uncommon situation. Actually, the week after that patient came in, this patient came in, another S310F HER2 mutation from a Foundation Medicine report. If you look at the pattern of mutations, it's completely different. The first patient has HER2 and NF2 and P53. The second has HER2, FGF receptor 1 and PIK3CA. Are they going to respond to single agent inhibitors? I really don't know, but it would be nice to test that in the context of a clinical trial where we can collect the data and actually figure that out. So we've opened up this study. It's just gotten open. We only have one patient on the study. This is protocol 13140. This is a basket study of neratinib, which is a selective HER2 inhibitor. There's 5 defined arms. You actually don't see lung and breast on this, although they're eligible within the other group, and that's because we have specific studies for lung and breast cancer open at the institution. So again, I think this approach really helps these rare diseases to a greater degree than other cancers. And what's notable about this study, we've actually amended it to now allow HER3 mutant patients onto the study, and we're also going to allow patients who have extracellular domain mutations of EGFR on the study based upon data from Ingo Mellinghoff that those mutations respond preferentially to lapatinib and neratinib versus erlotinib and gefitinib. And so this is the study, this is the assay that we're going to use to hopefully identify these patients. It's a next-gen sequencing assay. You're going to hear probably a lot of similar things about today, where we're going to test for 340 genes prospectively in many patients who come to Sloan Kettering. I'm not going to say all. So just an acknowledgement slide to just say this is the work of many, many people, and then I look forward to answering questions at the discussion. So thank you.
Thank you, David. So I'm going to take the liberty to ask the first question, and sort of an overview question to get things started. What are the greatest obstacles to realization of routine personalized medicine at this point, leveraging the genome to optimize every patient's treatment and outcomes? What stands in our way? What are the most important changes we need to make as a society to realize the health benefits of the technology? And you have to answer that in 22 seconds or less.
Is this even on? Okay. Well, I'll start. But I think the technology is there. We've actually, in retrospective studies, run our next-gen sequencing assay, which was developed by Mike Berger at our institution on over 3,000 cases. The sensitivity and specificity of those assays is incredibly high. Essentially, there was only one mutation during the validation phase of the assay that was missed, and it turned out it was a 30 base pair deletion, which could have been picked up. It was there. It's just bioinformatically was missed by the pipeline. And so I think that these assays are robust. I think they exist. I think the issues center more around reimbursement and throughput. I think that even if we wanted to today test everyone, I'm not sure there's the sequencing capacity or equipment in place to do that.
I think the single answer from me is reimbursement. I think that all of the other the technology to be able to examine all of these different types of changes is available. I think that there is capability to be able to look at as many cancer patients as there are available, that we could look at them, and I think that that would be forthcoming. Reimbursement is a big issue.
Well, I thought you'd all be running to the microphones. Oh, good.
Liz Mansfield from the FDA. Is the microphone on? I'm just curious if there were a commercial panel that could test for, whatever your 200 mutations or whatever you've developed at Memorial or any other institute. What do you think? I mean, a lot of those mutations are going to be useful for sending patients to appropriate clinical trials and won't really be actionable in terms of, oh, there's an approved drug or something like that. What kind of controls in clinical medicine would need to be in place for those kind of tests to be widely available that would kind of really send people to clinical trials that ought to be going to clinical trials and not being treated, sometimes a one-off basis is appropriate, but in a lot of cases we don't get a lot of information about that. So do you think that there are actually controls within the clinical community that would assure that tests like that, if they were widely available, would be appropriately used?
So I think it's a great question. I would say that the tests are already available and are commercially available. I think there's many commercial providers that are offering this test. I think that there's patients who are not sending their tumors, or doctors that are not sending the tumors to these commercial vendors in many cases because they're worried that the patient's going to get a bill that they're not going to be able to pay, and if nothing interesting is found, or even if something is found and something's tried and it doesn't work, the doctor's going to have, maybe put the patient in a financial hole. So I think the assays are already being offered, and because they're expensive, there's a risk that right now that these are only being, performed on patients who are wealthier, who potentially can pay for the analysis. But I think increasingly, it's being driven by patients, and they're showing up in our office with the report. And so Sloan Kettering might be a little bit of a biased example, but I actually think that these are being ordered more in the community than actually in Sloan Kettering, in my experience. So I think community doctors are ordering these, and patients are getting them. But the bigger problem is then, what do you do with the results? Can you get drugs? And to me, it's completely random right now. We've seen some examples where, based upon a case report, someone was able to convince an insurance company to provide a drug like everolimus or vemurafenib to a patient with a TSC1 alteration or BRAF alteration, respectively. But most of the time, they're denied. And patients oftentimes will be tempted, or their families, to sink tens of thousands of dollars into paying for these drugs off-label in examples where maybe there's 30 or 40 other patients that have already been treated and it's already been proven it doesn't work. So I worry that patients, for example, with colorectal cancer and a BRAF mutation are paying out of pocket to take vemurafenib, even though we know, actually, based upon really unpublished data, that it doesn't work. Even the Scott Kopetz abstract from ASCO like 4 years ago remains unpublished in manuscript form. So those of us in the know it doesn't work. It's somewhat public, but not in manuscript form, and so there's a real risk. And so how do you get around that? I think there's novel ways that we can develop trials to make these drugs accessible. So there's really no reason that a basket study like I laid out couldn't be more widely available than just a few institutions. There's no reason we couldn't make it available in a very broad geographic distribution so that patients go on the study, because I worry, as you do as well, that if patients are treated one-off in the community, you're not going to learn anything, and then we're going to just keep repeating the same mistake again and again if we have patterns of mutation where we don't see activity.
Thank you for a great presentation. So maybe this is a somewhat related question. I'd like both the panelists to comment on the speed of dissemination of the kind of explosion of information that's coming out of studies like the ones you've referenced. Is peer-reviewed publication timely enough and sufficient to spread this information as it should be disseminated to other researchers and including payers as well, who obviously probably could be guided by some of this information? And what other mechanisms are in play to ensure that this kind of data and research is spread widely and as quickly as possible?
So, you want to take that first? Or?
So I first of all, having peer-reviewed publications is really excellent because somebody has looked at them and the results are good results, and those results are actually coming at a very, very rapid pace. And the kinds of lists of genes that David showed, for example, or other types of lists that commercial entities are putting together are already based upon scouring of the literature and trying to determine the most significantly mutated or modified genes and looking at them. So I think that our ability to accumulate information and certainly this dissemination within the scientific community is pretty good. Whether we could disseminate to the rest of the community and be able to show that there is really clinical utility in doing all of these kinds of tests, and that's probably what's lagging at this time.
So, I think you're hitting on a great point, and I'm actually very frustrated that we haven't been able to disseminate the results better. So there really are a lot of unpublished observations that are not out there in public because the trial's not complete, and thus the data is considered confidential. And this is really problematic. I think the reason that the data is confidential has got nothing to do with the investigators. I don't think it has anything to do with the FDA. I don't think it's even the company that per se is restricting it. I think it really has to do with Wall Street and this concern of insider trading. And this is really problematic. We've let something that, there was potential for financial manipulation, so you have to keep these data confidential until it's reported broadly. We've let that reasonable concept actually restrict our ability to communicate data. So I don't know how to get around that. I think that there's maybe a way to have regular announcements about the data. This is something that I brought up with Puma, who's sponsoring the neratinib study. I don't think we should have to wait 2 or 3 years to present anecdotal data that starts coming up on this study, but they've already gotten feedback from their legal advisors that they have to be very cautious about reporting data in a non-public way in terms of they're a small biotech company and stocks could prices could change based upon these anecdotal reports. So I think we could come up with a way. Maybe there's regular releases of the preliminary data, but I think that we've swung a little bit too far on the pendulum in terms of secrecy for some of these studies, which makes sense maybe on a randomized phase 3, but not on a phase 1 or phase 2 study.
I might just add that there's one group of individuals who need this information, and this is what Harold alluded to. 80 to 85% of the cancer patients in this country are treated in the community oncology setting, and they're very busy practitioners and spend all the time caring for patients and don't have adequate amount of time to scour the literature and so on. And because they are the caregivers for all of these cancer patients, it's very important to be able to try to get the information to them. And the current modes, getting into the guidelines like NCCN guidelines or compendia, takes a long period of time to be able to get this kind of information. And there are other sorts of ways that people have been thinking about that as to whether we could provide them with the therapeutic pathways that would provide the latest information with the defining the quality of the evidence, nature of the evidence with level 1, level 2, like the ones that David described, provide that kind of information to them so that it would be available at their fingertips.
Can I speak to that?
I was going to.
I'm sorry. I'm Dan Hayes from University of Michigan, but this is an AACR meeting, but I'm going to speak for ASCO for just a moment, and it's relevant to your comments. So ASCO has just made a huge commitment to developing a rapid learning system. It's called CancerLink, in which we'll hopefully do just what you've recommended. Now, it's going to take a while, and it's a massive undertaking, but it will be providing guidelines, we hope from soup to nuts, and then gathering data from the clinical practices into probably the world's biggest database up to now, and then using that to rapidly modify the guidelines or rapidly modify the practice behavior. I mean, that's the overall vision. When and how that will happen is not entirely clear yet, but we have made the commitment. It's been very publicly announced now. And I completely agree with you. I think this is the way to go. I'm sorry to interrupt you. I just wanted to.
No, that's okay. Victor Valkoleski from Johns Hopkins. I had a related question, and I think it may partly have been answered by this discussion, which is obviously in great presentations. Obviously, as we're accumulating these type of data, they're helpful for clinical trials and for understanding, let's say, mechanisms of sensitivity and resistance to various therapies. But a lot of the individual patients that begin doing this will get data that may be helpful simply in guiding them to some trial that is already out there. And the question is, I think, obviously the folks that presented and the folks in this room may be very educated in this, but a lot of the community oncologists and a lot of the folks out there may not have this type of information at their fingertips. So how do we, what do we need to do in terms of education and trying to get this information out to as many patients as possible? I think part of what we've just heard may help in that regard, but I'd be curious as to any other thoughts.
So I think that there's a lot of efforts to try to create these computer-based tools that will direct patients maybe to potential trials. I think the problem with it is that the number of potential trials is not large enough to make that even worthwhile. So most of the patients who have interesting genetic alterations discovered at Sloan Kettering, there isn't a trial available for them. And the number of trials is very small right now that are pan-disease. And that's why I like the baskets, because any patient with a Her2 mutation is eligible. So we've taken that out of the equation. We've got lots of trials where we're doing a MEK inhibitor plus a PI3 kinase inhibitor, but you'd be amazed how restrictive they are. You have to have colon cancer that's failed one line of therapy. If you've had two, you're not eligible. And so patients might think they can take a rational approach, or doctors might try to give advice, but if patients are ineligible for the studies, it's not really happening. So I think we need to have studies open that have broader eligibility. We need to make the studies available to a broader range of people. And then we could have a lot of studies that really could be chosen from. But at Sloan Kettering, what do we have available right now? We've got a Her2 basket study. We've got an FGF receptor basket study. We have a BRAF basket study. And that is it. But what would I like to see? Well, a study open for TSC1 alterations, or an NF2 alterations that hits mTOR, a MEK inhibitor study for MEK mutations. We don't got that open. So if you find another patient with the exact same MEK mutation, my only choice is to beg, GSK to give them the drug. Maybe the insurance company, approves treatment, but why would they? I haven't even published this result. So I think that we need to have more trials that have broader eligibility available to make that even a worthwhile concept. Yeah.
I might just add something to David said, Victor, about the clinical trials. I think the patients that are in academic medical centers, 30 to 40% of them could end up in clinical trials. Whereas all of those patients that are seen in community oncology centers, only about 3% of them getting into clinical trials. So, and yet, there are so many drugs that are currently in development, and they're starving for patients from North America, for example, and they're not able to get adequate numbers of them. And part of that is, each of those practices, small hospitals or individual practices, are relatively small, and they don't have adequate numbers of patients to be able to get them enrolled, nor do they have the infrastructure to be able to do that. So one idea is to try to be able to build the networks of those groups, so together that they'd be able to contribute patients, and that's also a mechanism to be able to disseminate all this knowledge to that group of physicians and patients.
So we have 4 questions pending and 5 minutes, so let's have rapid fire.
Sure. I'm Dave Rim. I'm a pathologist, and I'm a member of the CAP Molecular Oncology Committee. And I'd like to follow up on Liz Mansfield's comments, and that is, how do you know you're right? One of the things we struggle with in molecular oncology is, even with EGFR, a relatively simple mutation to detect, 10 out of 200 sites tested got it wrong. How do we know that you're right, and what sort of, and the more time, and if you can each comment on the likelihood of having error as you have a higher number of genes that you're testing for and a lower frequency of mutation within the population you're testing, what kind of QC do you envision that would allow the 85% of the patients that are in these clinicals, in the private setting, to get the right answer?
First of all, I think the problem is not so complex, David. There's been a Nature paper that was published earlier this year. It shows the average number of somatic mutations that are present, in the tumors is maybe about 2 to 3 mutations per megabase. And if we look at all of the 300 genes that David talked about, or 400 cancer genes they look at, that they encompass, the coding regions would encompass about 2 megabases or so. So the number of mutations that you're looking at is not that high. So you could, and obviously there's going to be a range of them, where you'll be able to find the mutations. And if you could use criteria and be able to say that this mutation has to be in Cosmic or some other database to be valid. So it's possible to be able to determine whether or not the ones that you're looking at are significant or not.
So I'm gonna take a different approach and just say I think that it depends upon the clinical situation, and I think it's important to have very robust tests for things like EGFR, like 58R, where it's standard of care. But what I find frustrating is that there's no accounting for the fact that if you're doing an investigational clinical trial in the phase 1 or phase 2 setting, this is not an important issue. So if you're doing an investigational test to get someone on an investigational therapy who has end-stage disease and is absolutely 100% going to die of their cancer, whether there's a 1 or 2% false positive or negative rate is irrelevant. And I think it's usually the people who bring this up are people who don't see any patients. And I had clinic yesterday, and I transitioned a patient to hospice yesterday. And I don't think it's important to that patient whether there's a 1% error rate. That patient's going to die of cancer, whether they go on the drug or whether they don't. In all likelihood, their only chance is to go on the drug. And so I just think there should be a different standard depending upon the clinical situation, but there's no recognition of that right now.
Laura Van Veer, University of California, San Francisco. I had actually a follow-up question on that because I think you introduced us to the beautiful basket trials and also the battle trials, the adaptive randomization, but which is not available to everybody, yet there are more mutations found in patients, which sort of prompts physicians every once in a while to do off-label use of particular targeted drugs. So wouldn't it be a good idea to actually come up with a set of recommendations how to interpret or in what context to actually go to off-label use so that we can start to gather the data from everybody who is using those in such a way? Because there's a lot.
I think that there was a concept that we threw out there, which is, could you come up with a way to make a really inexpensive clinical trial design so that you could treat patients for a couple $1,000s per patient on a broad trial where all you were paying for was data collection, and the company would provide the drug for free. You wouldn't have any bells and whistles at all into the study. But really what you want to do, and the problem with off-label use is if patients are given the drug and nobody documents the experience, you don't learn anything. And eventually, maybe over a cumulative period of time, people do. But you have to have a way to collect the data. And unfortunately, data collection costs money. Many doctors in the community don't have an RSA or any clerical support to do that. So is there some way to do sort of an umbrella study where the patient, what does the patient get from it? The patient gets free drug. What does the company get for it? The company gets the data. What does the investigator or just the clinical guy in the community get? They get the benefit of maybe seeing their patient respond to this off-label use of the drug. But I haven't been able to find a way to get that approved, through the FDA, or get companies convinced that that's worth any money invested in it. But I think there's a way to do that.
I'm ready to help because I think it's needed.
And I think this has to be our last question.
Okay, Vince Miller, Foundation Medicine, thank you. So wow, a lot of meat on the bones here, but a couple of comments. One is to David. I think the issue is that our ability to do high-quality genomics that are broad-based in clinical practice has sort of outstripped where pharma is, right? So many of the FGFR trials are written for, an IHC-positive ligand or a test by FISH, and may miss fusions, may miss point mutations, et cetera, et cetera. So we really need to work collectively across our pharma partners to bring them up to speed. The other thing too is around publication. There was sort of early noise that there's a lot of this out there. If you submit a case report to the JCO, they're all backed up. They're not taking any more of those N of 1 cases. And that's sort of an interesting thing there. And at the lung cancer journal, the JTO, But I think we need to think about, when we talk about payers, the levels of evidence that we need, because it's different than what a blue ribbon panel, I think, around the room would think for some of these different things. You know, if we have small series of BRAF in 6 different tumor types with responses, knowing that two other tumor types were not efficacious for vemurafenib, nonetheless, that makes a good body of evidence around that alteration sort of in a pan-tumor context.
So I think it's a great comment, Vince. I think we got Dr. Varmus in the room here, and it's possible we need a PLOS N of 1 journal. The problem is if I suggested, he might make me the editor, and then I'd have even more work to do. So, but I think, yeah, we got to disseminate the data. And I think there's something you highlighted for these diagnostics. We've been doing our best to take ridiculous eligibility criteria out of clinical trials. That's been our mission over the past 6 months. And examples would be, we've got a Foundation Medicine report showing Her2 amplification, and the trial suggests that we need to repeat the FISH, which is ridiculous because the Foundation assay is more sensitive than the FISH. So I think it's just, you need to just look at these details and spend time with the regulators to improve the clinical trials so that you overcome these logistical issues.
Well, thank you, Raju, and thank you, David. Amazing what you got accomplished in 60 minutes to help us better understand the state of genomic understanding. Thank you.